I will tell you that the genetic testing of embryos is perhaps one of the more controversial aspects within the IVF industry right now. Can we make eggs and sperm out of stem cells? Can we genetically engineer embryos? There are many reports that suggest that sperm counts are decreasing.
You mean that 90% of the people, they won't be able to have a solution for them?
Yeah, the percentages and the way they are marketed by a given clinic are, I would say, widely deceiving. We call this still the Wild West. Unfortunately, I think the darkest aspect is the recent.
Thank you for being part of this journey to make genetics more accessible to everyone. In the past decades, how would you say that our knowledge or understanding of human reproduction and human egg fertilization has evolved?
Well, it has evolved significantly for someone who began studying the egg and its development within the ovary as a PhD student over 4 decades ago. It was, I think, originally a cell, especially for the human, that we knew very little about because we really could not study the human egg. So I would say at that time it took nearly 2 decades before the classical work of Bob Edwards began to explore human egg biology in a way that would eventually lead to the birth of the first test tube baby, and which took place, the birth took place the summer of 1978, but it was preceded by nearly a decade's worth of fundamental research on the basic genetics of the human egg, and specifically the genetics of meiosis. So we certainly understood at that point that the human egg was arrested in a very early stage of meiosis. We call it meiotic prophase. And it was really Edwards who was fascinated with the idea that these chromosomes that had been sitting in the ovary for many, many years would reach a point with development of the ovarian follicle such that when ovulation took place, this incredible process, which we now call meiotic maturation, took place to prepare the egg for fertilization and subsequent development of the embryo. Certainly, we don't have enough time to review all of the intervening history of our understanding of the human egg, but I can honestly share with you that IVF or ARTs really allowed us to begin to study the human egg in so many ways along a pathway where technologies moved from basic observational findings to technologies that allowed us to actually study the movement of the chromosomes, not only as the egg prepared for fertilization, this process, completing the process of meiosis, but now we know so much more about the early genetics, if you will, of human embryo development as a result of ARTs. There's no other way to put it, okay? So that's just sort of a brief survey, if you will, of how it started. And, as I'm sure many of your viewers know, one of the big issues with the human egg is that as women age, the egg becomes prone to making errors in the segregation of the chromosomes during meiosis. And these errors don't preclude fertilization of the egg. They don't preclude early development of the egg, but they can result in cases such as Down syndrome in the baby inheriting an extra copy, in this case of chromosome 21, which has serious developmental consequences and lifestyle consequences for children born with Down syndrome. And their families, of course, are impacted. So this age-related loss of let's call it normal meiotic genetics has been the subject of so much study, and since I know you're interested in how this projects into the world of ARTs and IVF, it no doubt has become a centerpiece for basic and clinical research because we now know that not only the human egg, but the human embryo after fertilization is subject to many fundamental errors in the distribution of chromosomes. So we now know, for example, that from, again, 2+ decades of research on human eggs that have been used in the clinics to treat infertility, that probably 80% of all human embryos have some form of genetic aberration. And how you get around this really, I would say, surprising behavior of human eggs relative to other animal eggs is, really a fascinating and very unresolved subject right now. And in fact, quite a controversial one.
And yourself, how did you get started to interest you in that area?
Well, the first half of my career, I was, really very fortunate to have trained in basic science within an academic setting. So, when I finished graduate school, and as I mentioned, that's when I really started to work on the egg and how it develops within the ovary. I had several faculty appointments as I worked up the academic ladder, and those faculty appointments gave me three really amazing opportunities that I think all have been woven together in terms of where my career stands at this point. The first was, of course, fundamental research, basic research into the human egg, truly the oocyte before it's mature. And so funding from various governmental agencies like the National Institutes of Health, March of Dimes, a number of groups were interested in the kind of research we were doing and funded us for almost 40 continuous years. And in parallel with that, at least for academic institutions in the United States, we were, as junior and then senior faculty members, expected to apply for grants to support our research and and also contribute to teaching. So I think one of the very fortunate experiences I had over the early phases of my career was to be a teacher, to be an educator, and an educator within medical schools. So over the course of my career, I have taught anatomy and physiology at 5 different medical schools. And, I was always put to task to teach in a way. In those days, it was mostly lecture teaching that would stimulate medical students to not only understand complex issues, but raise their level of curiosity so they would want to explore each subject that we covered on a deeper and deeper level. And I honestly I look back at the high expectations that the institutions that I worked within had for excellence in teaching as something that contributed to my own career development in terms of writing papers, communicating with other professionals in the field. And, to that point, I've been fortunate to use those skills of communication, both in the written form and in the spoken form, as an editor of a major journal in reproductive medicine, the Journal of Assisted Reproduction and Genetics. And I'll just add that having those communication skills and trying to understand very complex problems such as the biology of the egg cell or the biology of the embryo really has, benefited from those earlier teaching experiences, and which I got to share with many PhD students who trained in my laboratory, with many postdocs, and most of all with collaborators that I've had from around the world. And I attribute a lot to that. Now, that all said, there's actually a third element of my career that is probably representative of many career scientists in the United States, which is you're expected to do research and find money for it. You're expected to teach, and if you're once you become good at those things, then your institution is likely to ask you to become an administrator, a department chair, or a dean, or this sort of thing. So that that has happened a couple times in my career as well. But I love what I do. I think the egg is the most fascinating cell in the body, and there's certainly nothing more important than understanding human reproduction. And certainly, as my transition as a basic scientist moved into the clinical field, I came to appreciate assisted reproductive technologies, what was working to help patients have a family, and most importantly, what was not working. And so over the last 25 years, I've been very involved with clinical work, and I've, come to understand some of, first of all, understand the very basic biological principle that human reproduction is very inefficient. I learned this from my very first mentor, who in the 40s did studies on human reproduction, which taught us that in fertile couples, so healthy, young, fertile couples who were
Yeah.
able to have families without any intervention, that only 1 out of every 3 fertilized eggs would ever make a baby. Compared to the other mammals that we studied, rabbits are 100%. Okay. Every time a rabbit mates, there will be a rabbit baby produced. Okay. And humans are at the other end of the spectrum. Humans and also subhuman primates, we've come to understand. And this has been one of the big challenges in the field of assisted reproductive technology, where we have to ask, even with its low efficiency, which we can return to, can we do any better than normal human biology? And I think the answer at this point is no, we don't do better, but we are fortunate enough for couples who need interventions like assisted reproductive technology, egg freezing, embryo freezing, all of the things that have now entered this field. We at least in, let's say, 10% of the cases can help couples realize their hopes of having a baby and starting a family.
I have many questions. You mean that 90% of the people they won't be able to have a solution for them?
Yeah.
The people who are looking for a hierarchy?
Yes, let me dig a little deeper into that number, because it's probably surprising to you, and it would be certainly
Yeah.
surprising to most couples who maybe have tried to conceive for a year and they now are going to seek out professional medical assistance to try and have a baby. The 10% comes from a series of studies of IVF clinics in the United States, and this probably relates to IVF clinics around the world, where a fundamental question was asked. Of all the eggs that we retrieve from women, and we retrieve more than the normal one egg that is ovulated every month, we stimulate women so that they produce more eggs. So if we look at a very typical patient, maybe would be a good way to think about this. A 33-year-old woman comes into a clinic She does have regular cycles. She and her partner have been trying for 1, 2 years. She goes through a medical workup to see if there is a definable reason in her own biology for why she could not get pregnant. And so she's advised to enter what we call an IVF cycle. And that cycle will involve having her inject a series of hormones at the beginning of her menstrual cycle, monitoring her ovaries to see that many of the follicles, each of which have an egg, are developing. And at an appropriate time, the woman will take an injection of a hormone that will induce ovulation. And again, thanks to the original work of Bob Edwards, clinicians now know that 30 to 34 hours after that injection, the eggs within that ovary should have matured and should be ready for fertilization. So let's take as our starting point this patient who yielded 20 eggs. Okay? And let's assume for the moment that all of those eggs were gauged to be mature and ready to be fertilized. So now, with a technique called intracytoplasmic sperm injection, or ICSI, we would take individual sperm cells and inject them into each of those eggs. And 24 hours later, by looking at that egg under the microscope, we would decide if the egg had been fertilized normally or not. And again, for the sake of this conversation, let's say that 24 hours later, all 20 of those eggs appeared to be normally fertilized. They're then cultured for some number of days, typically now for 5 days, and we monitor how the embryos look. And reality is that perhaps half of those embryos, let's say 10, would have developed to the blastocyst stage, would have presented all of the characteristics that we know are indicative of a chance for that embryo to implant and a chance for that embryo to go to term and produce a live birth. So with those 10 embryos, we can begin to ask, as these studies did, what percentage of them ever result in a live birth or a baby? And these, three, I think, seminal contributions to the literature that I must admit are very difficult for the field to accept. Those studies have shown that only one out of those 10 embryos will result in a baby. Now, mind you, we just created kind of an ideal scenario with a young patient that made many eggs, of which all of them fertilized. That in itself is not reality because, as for example, as patients get older, they don't respond as well to the hormones. So instead of getting 20 eggs, we might get 10. And when we fertilize those, only 5 would be able to develop. Okay? And then of those, if 1 out of the 5 developed, it would be wonderful. But typically in older patients, none of them develop. And so patients are required or requested to go through multiple cycles so they can essentially stockpile their embryos in a frozen state and again revisit this question of if I've accumulated 20 embryos now in a frozen state, if we thaw those out and begin to transfer individual embryos, and that's important because the field right now, we use what is called single embryo transfer, as opposed to a decade ago when we might be putting 2 or 3 or even more embryos in. But if for that patient, the older patient who had fewer eggs to begin with, but went through multiple cycles, again, we would see on the order of only 10% of those would have ever produced a baby. Many of these, by the way, are transferred. The embryo implants, but some weeks later, very often there will be a miscarriage, a loss of the pregnancy. And again, we know that to be the case in normal, healthy couples, which is why those classical studies originally indicated only one of three fertilized eggs really ever go to term.
And you made a comparison with rabbits. Why is human reproduction so inefficient compared to other mammals?
Yeah, yeah. It's a fascinating question. And I would say we don't have answers to that question, but there are many hypotheses about this. And they go all the way from a fundamental molecular genetic level to what I would call a societal level. So I'll eventually end up sharing with you what is called the grandmother hypothesis. Okay. But where this starts is asking, for example, what's so different about a rabbit and a human with respect to their reproductive strategy? Well, first of all, a rabbit is among species of animals that will only ovulate if it mates. So what this means is that the rabbit is in a state of readiness, ready to release 10 mature eggs exactly at the time that the fresh sperm are introduced into the reproductive tract. So there is, in a sense, let's call it perfect timing between the viability of the sperm and their ability to get to the site of fertilization and the viability of the eggs. So ovulation is synchronized with reproductive behavior, with actual sexual activity. Okay? So if we were to take that dimension of reproductive physiology and ask, well, how does a human differ? Okay? In many, many ways. Okay? We think that ovulation occurs within a narrow window of the menstrual cycle, and we certainly understand what the hormones are that prepare the ovary for releasing that egg. And in normal human reproduction, only one egg will be ovulated. Of course, sometimes two in the case of fraternal twins, okay? And as far as sperm entering the reproductive tract, are you on time? Are you a little too early? Are you a little too late? Okay, so there's no guarantee that the sperm that have the competence to fertilize an egg will be in synchrony with the age of the egg at ovulation. And in fact, we know that when a human egg is ovulated and it's in this mature meiotic state prepared for fertilization, that that state exists for a narrow period of time. So the human egg, for example, is fertilizable, we say, within probably 14 to 16 hours of it reaching its fully mature state. Now, if an egg is aging inside, is it still fertilizable? Absolutely. So if, for example, a woman ovulated on day 14 and sperm were not introduced into the reproductive tract until let's say day 16 or 17, that egg could still be fertilized by those sperm. But because it is already aged, it has lost both the machinery and the capability to undergo normal fertilization and more likely to develop. In other words, the egg will fertilize, but the embryo will be abnormal in its subsequent development. And in those cases, since the embryo never gets to implant, all a woman would experience would be a delayed period. Her cycle would be extended for a little bit of time, a few days, only because there was some signal coming from that aged embryo that was starting to tell her, oh, you might be pregnant. So it's not time to menstruate. So some days later, that embryo will die in the reproductive tract, and then menses will commence. So that's at a fundamental biological level. Now, what about a molecular genetic level? As I mentioned before, this is a very sophisticated process for the egg to get its chromosomes in order and separated equivalently to produce what we call is a haploid gamete or oocyte. By the same token, the sperm carry a haploid genome, and when the sperm fertilizes the egg, these two haploid genomes need to first undergo replication of their DNA, and then these genomes essentially intermix with one another, presumably now constituting a new set of 23 plus 23 chromosomes, 23 from the female, 23 from the male, constituting a new and uniquely genetic zygote, which will carry the properties of the genes on each of the chromosomes from mom and from dad. Again, if we take an animal model like the rabbit and try and put that into context, what we know is that this process of replicating those genomes and getting them to mix and launch development so the embryo will divide is very efficient in mice and rabbits, okay, and hamsters and so many of these other animals. But it turns out that, and again, thankfully, we have been able to research these problems because of the whole field of IVF evolving over the years. Now we know that even that very first step of intermingling the chromosomes and separating them correctly is error prone. Remember, I mentioned that there are issues with the human egg anyway as a woman gets older. These
Yeah.
these errors tend to be more common as women age. Well, the same is probably true for men and the sperm they produce. And certainly we know now that there are some very peculiar, in fact, risky behaviors that we see in the way that the cells of the embryo go on to divide through those first 3 to 5 days of development. So much so that, by testing the genetic composition of some of the cells of the embryo, as I mentioned before, we're kind of coming to accept the fact that perhaps 80% of all human embryos have some form of a genetic error that they're carrying. And, again, remarkable that it works for us. Okay. So that's two levels, kind of the genetics, the basic biology. So let me share with you the grandmother hypothesis. The grandmother hypothesis argues that a woman's most fertile years are in the teenage to early 20 years, where the woman alone has sufficient youth, energy, is able to produce milk to feed the baby. This is a little bit more of an evolutionary way of thinking, such that survival of a child that's born is more likely to occur with an attentive mother and one whose physiology can match the nutritional demands of a developing baby. And in addition, we have to put in the framework that of the various species on the planet, excluding whales and elephants, okay, that humans have a very long gestational period. It takes 9 months for a healthy child to complete its development in utero and land on the ground with sufficient maternal support to proceed through the early stages of development. So there's the long gestational period. And again, going back to the rabbit is very short. Okay. In 20 days, the babies are born. So shortly after 20 days, those mothers can go into what's called estrus or heat. They can mate again. They could have more babies. Okay. So just if you will, the chronology of reproductive cycles also distinguishes the human from other animals. But more importantly is this idea that younger women, have literally the energy resources to invest in their offspring. And of course, if they're lactating and nursing, what is the biological effect of this? It is to suppress ovulation and make sure she doesn't get pregnant again. Okay, the last thing she would need is to be like a mouse or a rat with a litter coming every 20 days, and suddenly you have too many offspring to care for. Okay, so there are consequences of this that suggest that the reason that, let's say, pregnancy loss, miscarriages, abnormal chromosomal embryos are seen later in a woman's life is because this is a mechanism for ensuring that 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 women have a decreased reproductive capacity with age, so as not to jeopardize the investment of her energy and attention in the development of young. So where does the grandmother hypothesis come in? The grandmother hypothesis comes in as an accommodation of this trend of losing reproductive competence over age, because if there's a grandmother around, she can actually support the mother and provide, nutritional support, psychological support to again now assure that that generation of offspring reach sexual maturity and themselves acquire reproductive competence to add to the propagation of the species. So mind you, especially this last idea, this is, it's very hypothetical. It has sociological implications. And it's frankly one of the reasons why IVF and assisted reproductive technology is just not as successful as we would expect it to be in a 24-year-old woman who has a partner producing high quality semen to get that process going. And as I alluded to, both the aging of the male now we recognize is contributory to the failure in IVF cycles to some level. And as we know, as women age, the quality of their eggs diminishes, and therefore the chances of producing a healthy embryo diminish and taking home a baby. This was really the primary incentive for the egg freezing industry and egg banking, was to give women an opportunity to store those eggs when she's at a younger age and come back to them when she was ready to start a family at an older age. Because the rest of the reproductive tract, and we know this now from oocyte donation studies, the rest of the reproductive tract, such as the uterus, which needs to support implantation of the embryo and sustain via the placenta the development of the fetus, the uterus still works pretty well. So the problem in older women having more miscarriages, pregnancy losses, etc., is not one necessarily of the reproductive tract as it is of how that egg cell developed in the ovary and predisposed it to, these genetic problems, chromosomal aberrations that can compromise development.
In addition to age, is it true that nowadays the quality of semen of eggs has decreased in the last decades?
Yes. And we speak to both sexes when we talk about this now, right? And, so for example, there are, many reports that now suggest that the quality of the semen, the sperm counts in men. I think many of these studies came out of the Scandinavian countries initially, that sperm counts are decreasing in current male populations when compared to sperm counts 30 years ago.
Yeah.
Okay. And that's a quantitative measure of, if you assume that more sperm is better than fewer sperm, then yes, that would lead to a decrease in the reproductive capacity of men. And we can add to that conversation right now lifestyle factors, environmental factors, and nutritional factors, that these are all at least on the table for discussion right now. But we don't know which of the culprits is the worst, and most likely it's going to be some combination of those factors, including genetic factors. Okay, we're with the new world of molecular genetics, we're understanding much more about the actual genes that control the fertility of men with respect to sperm production, and the same in the female side. So there's a whole world, I think, of genetic diagnosis and genetic insights that we've yet to really, fully seen mature to incorporate into our thinking. Now, as far as the female side, yes, there's kind of the intrinsic biological aging problem, and we're certainly coming to realize why that is the case, and it has very much to do with the environment that the egg develops in within the ovary. So long before the egg would be ovulated, it is probably as a result of environmental factors. We know that hormonal factors change. That is, the dimensions of hormonal changes in the menstrual cycle are also changing as a woman ages. And to maybe make a simple metaphor of this, in women, the reproductive cycle, at least one part of it, actually gets shorter and shorter. So there's less time invested by the mother in supporting the nutritional requirements of the egg as it develops. So the egg will go on, it will be ovulated, it may in fact fully mature in many cases, but it hasn't acquired all of the maternal signature that makes for a good egg. So again, it could fertilize, etc., but it would be unlikely to support a sustained pregnancy. And again, you could superimpose on that line of thinking. You could superimpose environmental factors, lifestyle factors. Of course, there's a huge literature now on the impact of obesity. Okay, the impact of obesity in women, not only on the endocrine profiles, but also the impact of changes in a woman's metabolism that indirectly probably influence the development of the egg. There are, disease states in women that we know also compromise their reproductive potential. Probably, the two biggest ones at this point are polycystic ovary disease, which affects a large percentage of the population. 20 to 30% of women experience this at some time or another. And, as couples wait longer and longer to start their families, other health issues come up: endometriosis, fibroids. There, And since these are more likely to occur later in a woman's reproductive lifespan, then they're all, very real pathophysiological changes that can impact the ability of a woman to conceive and support a pregnancy.
And when we look at the solutions and the technologies for helping people to conceive a baby, we have spoken about freezing the eggs, for instance, of a woman when she is still young.
Hmm.
What are the different kind of principles of, of principles that exist
Yes.
or you think will exist that could be complementary?
Okay. Yeah, there's a very solid history of what we collectively refer to as cryopreservation technology, which was certainly, used for, you know, preserving other body parts and cell types, like blood cells and things like that. So the field of cryopreservation is really founded on the biophysical properties of living cells. And I think the three important cells, if you will, that we are talking about today are eggs, sperm, and embryos. Okay? And if you care to go there, I would be happy to share with you a fourth and very exciting principle, which has to do with actually freezing ovarian tissue and preserving the population of eggs that a woman could have used in her life. And this has evolved out of how can we help women in particular in this case who are going to be going through either chemotherapy or radiotherapy for a cancer diagnosis, because those treatments often will cause sterility of a woman because they damage the ovary. So back to cryopreservation and cryobiology, as we call it. If we, wanted to look at the recent evolution of this technology, I think what we could say is that before 2012, there was a technique developed first with animal eggs, mouse eggs, cow eggs, etc., where those mature eggs, the state they're in at the time of ovulation, when they're ready to be fertilized, how to preserve that state and viability was addressed by really understanding how to prevent the formation of ice crystals within the egg cell. Okay? So if so, and most people generally know this, our bodies are composed of 95% water, okay, as are our cells, okay? And so if you simply take a cell like an egg and you subject it to low temperatures, and we're talking about super low temperatures here, like liquid nitrogen, okay, at minus 221 degrees centigrade. So if you just begin to cool a cell down, that will change the ability of the cell to retain the correct amount of water and ions in the cytoplasm. And as you cool it to freezing temperatures, what happens is ice crystals begin to form in the solution that the cells are in, but they also begin to form inside the cell, in the cytoplasm of the cell. And if you've ever seen ice crystals form, you know they look like deadly swords, weapons.
Yes.
Okay. And so early on, I think it was realized that when ice crystals form, they damage all of the important ingredients in cells. And in oocytes, you've got mitochondria, you've got lysosomes, you've got the nucleus. All of these things were not recovered. So the mentality really of the biophysicist was, how can we replace the loss of water with something that will prevent or limit ice crystal formation? And so this is where the idea that as you use antifreeze in your car, okay, to protect the fluids in your car from freezing in cold weather, Many of these compounds, like sucrose, glycerol, DMSO, etc., have been around. They're organic compounds. They permeate through the membrane of the cell. They displace water. So now, as you subject the cell to these cooling temperatures, you limit the amount of ice crystal damage in that cell. Now, that technology was really the first one. In fact, if I'm not mistaken, the first case of a frozen human egg producing a baby probably goes back to the late 80s, early 90s, okay? But up to, I would say, about 2011 and 2012, that technique, which is called slow freeze, cryopreservation of eggs, required not only the correct set of solutions that you would put the egg in, but a machine that very slowly decreased the temperature of the solution all the way down to minus 200 degrees. Okay, so slow freeze technology was the first adopted technology to freeze eggs. It was quickly adopted to embryos as well. Okay, and sperm's kind of another story, so we'll stick to the egg and the embryos for now. Because a second technique began to emerge between 2005 and 2010, which is called vitrification. And again, this is also a technique that allows for the replacement of organic molecules in the cytoplasm of an egg or an embryo, and then a more rapid change in temperature induced by plunging the sample into liquid nitrogen. And essentially, in vitrification, the temperature transition change is so rapid that rather than the water molecules forming ice crystals, they form something called a glass state for the water. So it immobilizes it before crystal formation. And you can think of it as kind of a solid medium, and we use different cryoprotectants or different chemicals in the case of vitrification. And that frankly became widely accepted as an alternative technique for cryopreservation of eggs and embryos. Now, that all said, something very interesting happened. I believe it was a meeting I attended in 2013 when two schools of thought had emerged. One was a school that promoted slow freeze, cryopreservation of eggs, and the other was a school of people who promoted vitrification. Now, slow freeze, if you remember, required a machine, not inexpensive, okay? And that would mean that every laboratory would have to have this machine in order to freeze their eggs that way. Vitrification, much easier, much less expensive, and without going into the weeds of this controversy at the time, the American Society for Reproductive Medicine, which had previously decided that nobody should be freezing eggs for making babies, changed their mind, again, based on not only clinical reports with human eggs, but many animal studies. And in that moment, again, this was, I believe, in November of 2013, a whole industry of egg banking resulted. There was no such thing as egg banking before that, except in the occasional clinic that was really proficient with these cryopreservation techniques. And of course, for patients who want to preserve their eggs for use later on, this has become a wonderful opportunity and an alternative to waiting until your eggs are too old. For many patients, older patients, we now can offer patients whose eggs just are not performing well the opportunity to pursue egg donation. So they can purchase human eggs that have been frozen at one of these egg banks. Typically, the egg banks draw on younger women who go through repeated IVF cycles to collect and bank and accumulate many eggs. And if you want to talk about genetics, okay, this has become a big industry, right? For people who can afford to purchase eggs from that blue-eyed, blonde-haired, Ivy League graduate, okay, who would, for a certain family, bring them all of the desirable genetic traits that they would want in their offspring that they otherwise could never have realized. Okay. So I don't want to leave this conversation without sharing with you. There are huge ethical implications and complications in the way this whole field has evolved. As you and the viewers may come to realize, 20 years ago, IVF took place mostly in academic medical centers. Okay, it's a huge commercial industry now. Those academic centers are few and far between now. Where you would have a team of scientists and clinicians working to develop the best strategy for a given patient. Now you have the business incentive of how many patients can we get in here, how many cycles can we run, because the profit margins are huge for investors right now. They also, as you may or may not know, reproductive medicine specialists and fertility specialists are amongst the highest paid physicians in the world now. So that needs to all be contextualized appropriately.
And precisely for regarding that, for a couple looking help to conceive a baby, what kind of criteria should they use to select the best clinic to help them?
You're asking if you were a patient planning
Yeah.
to go through this, how would you identify and possibly be treated by one clinic over another one? This is a very dark and disappointing subject. And I say that because there has been so much false marketing by clinics as to their success rates. Okay, let's say we go online today. Okay, we're going to search. I want to look at 10 different clinics. I'm in the United States, so we'll look at 10 in the United States. And you will pretty much up front see, we have the best pregnancy rates, we're better than everybody else, okay? And there'll be numbers marketed. 50% of our patients get pregnant, okay? And that to someone, who sees maybe somebody's saying 20%, maybe someone saying, which is rare, that, oh, by the way, only 10% of all of our fertilized eggs make a baby, right? You would not be attracting patients if you marketed that statement. Yeah. So the percentages and the way they are marketed by a given clinic are, I would say, widely deceiving. So at that point, you have to go a little deeper, all right? So you may go in for a consult, and you'll go through some testing. At one clinic, you'll sit down with a physician. They'll start telling you about your chances depending on your age, depending on all of the various measures that are typically taken in an intake case. A new couple shows up in the clinic. And you say, okay, this, it sounds pretty good. They'll tell you something about how successful they are, generally speaking. And usually that conversation is a little tainted because many clinics we now have come to understand will share with prospective clients numbers that refer to only their patients under 35 years old. Okay? And in fact, There are large businesses in the United States who will not even entertain you as a patient if you're older. And the reason that they won't take you on as a patient is because they know success rates are going to be lower and they don't want the word to get out. They would much rather market and pre-select. This is a classical case of bias in clinic reporting, okay? They will only report what looks good. That's the best way to attract patients. So getting back to the real world scenario, you might go through a workup with one clinic and then maybe take your data to another clinic and ask for a second opinion. And this I think, I'll tell you, our patients are so savvy, they're so knowledgeable, there's so much information now on the internet, on social media. The good news is that there is a lot of information out there, and patients come in very, very well informed about things we've been talking about today: biology, genetic testing. How do they're really tuned in to the basic biology and the clinical results. Okay? So when you begin to sit down with them, I think come to appreciate that this is a huge financial investment, and the patients themselves, I believe, are becoming more discriminating. And unfortunately, I think the darkest aspect of picking your clinic, etc., is it's called the story of the add-ons, okay? Which means that there are many techniques that clinics will offer their patients at cost, and the physicians or the companies marketing these add-on procedures will claim better results, better outcomes, more frequent pregnancies, without any fundamental validation, basic research that would allow those statements to be supported. It's sad, but it's true. And the add-on controversy is something that I would urge most patients to very carefully become familiar with, and to have that in mind when they approach clinics. And we really don't have time to go into the array of them, but I will tell you that the genetic testing of embryos is perhaps one of the more controversial aspects within the IVF industry right now.
And regarding the ethical issues related to fertilized reproduction, what would be your current view on the laws in the USA regarding research, regarding clinics? Are there some laws that you find are not efficient, or other laws you would add?
Okay, well, that's very easy to answer. At least in the United States, there are no laws, no regulations. You can do whatever you want. If you and I wanted to establish a clinic today, we would pick out a state that wouldn't care. It would just be a new business entity. We would probably, if we had some money to invest, we'd hire a couple of doctors who do this and other staff members, and we could just start treating patients. Okay, now it's not quite as simple as that, but I think when it comes to the ethical issues, this is probably one of the larger voids in the relationship of a patient to a medical discipline. I am not an ethicist myself, but I read as much as I can about this. I have a excellent group of ethicists on the editorial board of JARG, who we often refer these issues to. But I think the lack of any regulation is one thing. Now, that said, The United States is very different from many other countries. Germany has very strict laws and rules. Okay? Most of the European countries have some form of legislation that's been passed to offer guidance for essentially fair treatment of patients and oversight for the clinics to make sure that they are, in fact, following not just guidelines, but following procedures in the best interest of the patient rather than in income generating opportunities for the clinic's owners. Okay? I would single out the UK and the Human Fertilization and Embryo Authority, HFEA, as really the model country that recognized the need for this interface between medical practice, legalities, and ethics. So this embryo authority has to approve any and all research projects that might involve using human eggs or embryos. It also provides oversight for the clinics. It has spoken directly to this issue of add-ons and at least imposes some level of information for patients to use in choosing the clinics that they want to go to, gives them guidance on how to think about this. But in the United States, we call this still the Wild West. It's gotten, I think, much worse from a patient perspective now that it's no longer a discipline in medicine that is really regulated in any way. So that's the best I can do without speaking. Yeah, yes, there are specific hot topics going on now. Can we make eggs and sperm out of stem cells? Okay. Can we
Yes.
genetically engineer embryos to eliminate a disease or to, make every offspring 6.6 taller? Okay. Blue eyes. Okay. But, fortunately, most of these technologies are, while there are companies promoting these technologies, most of them have not come to fruition in any clinical clinical way. Although there are certainly many inherent dangers to applying some of these technologies, like CRISPR-Cas9. Let's pull out a disease gene, put in a good one, that sort of thing.
In your career, what is the most interesting or the most surprising thing you discovered in your work?
In my own work,
Yes.
okay, all right, yeah, I hate to say it, but there might be two things, okay, that might be unrelated, okay? But I'm actually in the midst, I've been asked to write a special paper for, one of the high-ranking journals in our field. And it's about sort of my career interest in how the egg cell talks to the cells in the ovary and why that communication pathway is so critical to producing eggs that can form a baby. So many years ago, we discovered kind of a structure at the interface of the egg cell and the cells within the ovary. And we were able to use transgenic technology to make mouse models of infertility and, really came to understand much more about how making a good egg really depends on the cellular environment that it grows in the ovary. So if the cellular environment is compromised, it's very likely, as we discussed with aging, that the quality of the egg that's produced from that environment will also be affected. And so now there are a lot of people, not me, but people who have been able to build on this fundamental idea and are really trying to improve the quality of eggs that we get clinically, thinking about this very mechanistic interplay where essentially the metabolism of the cells in the ovary is being shared with the metabolism of the egg cell. And if that sharing is not effective, then very likely those eggs will not develop appropriately or produce an abortion. Okay. I think the second thing has to do with work that we did many years ago on drugs to prevent the growth of cancer, of all things. We did some studies many years ago on a drug called Taxol, which is still used to treat many forms of cancer. And the long and short of that is that as we were speaking of why does the aging egg make these mistakes in chromosome segregation, why does it appear that some of those properties actually go into the embryo and might underlie why there are so many chromosomal errors in the human embryo after it's fertilized. This has had to do with studying a specific structure and a specific molecule, my favorite molecule, which is called tubulin. And the tubulin molecule is what makes the structure that the chromosomes have to attach to, and it regulates how those chromosomes are segregated. So in an ideal world, when the chromosomes are segregated equally, we generate what's called a euploid or chromosomally normal cell. If the chromosomes segregate unequally, such that maybe one cell gets an extra chromosome and the other cell is minus one chromosome, then you create a state called aneuploidy. And aneuploidy was long ago recognized to be a very bad thing and suggested to be a cause for cancer. And so we really contributed classically over the years, not only in the animal system, but we focused on this when we developed some of the cryopreservation technology for human eggs. And we published several very important papers studying the effects of cryopreservation on this structure. So I'd say those are kind of the two things I'd be most proud of. And yeah, and thanks for asking me.
And also, I think one interesting aspect is that you worked in a scientific journal. And more broadly, there have been some critiques on scientific journals regarding their power, because they act as a gatekeeper for deciding
That's
what
right.
will be published, and they
Yes.
have a lot of power in that field. What would be
Yes.
your views on that system, on how research is done, and the relation between researchers and the scientific journal?
Yeah. I'm not sure how much more time we want to spend on these issues. That's another very large question. And as you mentioned, I've been very involved with scientific publishing for my whole career, but for over 16 years now, I've seen the whole medical publishing field change with respect to the journal that I'm editor of. We're facing a lot of challenges now, I would say. There was a time when scientists and clinicians really followed good principles in study design, in sort of, doing controls for their experiments, okay, and, asked simple questions that often gave reasonable answers to whatever the problem they were studying. Why is the human egg so prone to chromosomal separation areas? Okay, and and I think I see a real issue in the integrity of, certainly I would not want to make a generalization, but I see more and more papers coming through that lack the same kind of integrity, thoroughness, novelty that really changes how clinicians should be practicing in this area. I've made it a specific point of the journal I work with to try and bring that to the attention of our readership, okay? Because there are a lot of papers that get published that are very misleading in their take home message. If you dig deeply into them, you find out that their control populations are really not adequate control populations at all for the study design and the question they're answering. So I have to say there's so much more noise in the literature that's published right now. Now, there are a number of reasons for that. One of the ones that we as editors confront all the time is finding enough expert reviewers within the peer review process to form an informed decision about that manuscript when it's evaluated. Okay? Are the problems, so severe that the manuscript should be rejected? Do they require revision? Okay, and even if they do require revision, have the authors really addressed the concerns that were raised in the original paper? Okay, so often now we see submissions which have also increased by a log order in the last 10 years because of ChatGPT and everything. Then there are companies now that you can buy your authorship on a paper from. Okay, but we see so many papers still where they want to publish a study that they say, oh, this is new, this is eye-opening, this is going to have great clinical implications. And in fact, there have already been 20 very good papers on that subject. Okay? So, and that's just one of many examples I could give you about what I would say are real challenges in the medical publishing industry, and of course in how we relate good scientific findings, how we move them from the bench to the bedside, how we move them from a fundamental knowledge base into a clinical reality. And those challenges are going to persist for a while. And no doubt the introduction of artificial intelligence, large language models, etc., is throwing another major concern into this puzzle. And, the tendency, at least in the field of reproductive medicine, as I mentioned with oocyte cryopreservation, it was non-existent. And then as soon as a professional society said, oh yeah, go ahead and do it, it became an industry. And it is abused in many ways, okay? I see the same thing happening right now with artificial intelligence, large language models, these various dimensions of AI, at least in reproductive medicine. Every week we get 3 or 4 papers submitted that say, oh yeah, AI is going to fix all our problems. Everybody will have a baby because now we can extract some information from a movie of an embryo and tell you what its potential is for making a baby. And it's terrible.
So thank you very much for your time. We have a bit overextended. Is there anything you would like to add?
No, I hope people appreciate what you're doing in terms of reaching out to especially people like myself and some of the others I've seen you interview who cast, really a career broad view of how a field, in this case, like reproductive medicine, evolved over the years. And for me, I only got into science for one thing, and that was because I believed that if I could discover things about how cells worked, that maybe someday people would benefit by it. Okay, they would benefit in the sense that it's something that we published might become just a tiny fragment of a eye-opening concept that could, in my case now, it's obviously to help infertile couples, also to help couples manage their reproductive potential. But I feel that that's sort of the core principle that's guided my career, and it will continue to be.